So I told you about, we've already folded the DNA into the cell, looked at the conformation. For those of you who know a little bit about chromosome dynamics, this has what they call a more fractal geometry. It is not so interwoven. So you expect in the experimental C-maps, the 3 C-maps, that you would only see a single diagonal. If you had a mixing of the 2 arms with one another, you would see beginnings of a secondary diagonal coming in here. And if you have loops, you have to really zoom in. This is at about, I think, 1,000 base pair resolution, sort of these green spots. And if you zoom in on the in silico map, you can see there are things that are interacting with one another that are sort of what we call in loops. But in general, there's very little persistent supercoiling in the system. And this is in part because in the minimum genome, in reducing it, they also knocked out most of the proteins that are known as nucleoid associated proteins that help stabilize the DNA structure. And there's only about 1 left, and it's only 28 copies of it compared to over several thousand that are in typical other pathogens and mycoplasmas. So we were happy that it agreed with the chromosome capture map, but it does allow us to put a more open DNA structure in there and not have to take into account the dynamics of coiling and uncoiling to make a gene accessible or not. Now, the program to generate this DNA is all made available through that Frontiers paper, but I would strongly suggest, because it takes a while to generate an ensemble of structures. And when we do the 3D simulations, we sort of stay with the ribosome distribution, but we do replicates that have different DNA configurations in it. So no gene is only seeing a position near the membrane or in the interior of the cell. But they do take a while to generate, like a day to generate an ensemble of them. But we're working on a continuum model that people might want to use later, but that will only become available, say, at the end of the year. All right, so now we've got the cell geometry from cryo-electron tomography. We have put in that means the boundaries, the ribosomes, place the DNA, and we load in the rest of the proteins. Here I was showing you another example we've done on E. coli. This tomogram came from the lab of Wolfgang Baumeister. And again, this is for E. coli, but you see very clearly the boundaries, and you see a lot of black dots and a faded region here with no dots. So that means that when you, in this case with E. coli, the DNA was condensed and the ribosomes were surrounding it at the poles and the sides. And then we have an algorithm that places the proteomics in there, all right? So this is a packing algorithm called, I think, C-Builder, and it is written in a Python to help you do that, to set up the system, starting with placing the big guys first and then the smaller particles. Okay, so come on. Now, just to give you an idea how helpful VMD was when we were first studying these cells, here's a little movie we made on E. coli. It was done by John Cole in my group with the help of John Stone. And we had from CyberCell the distribution of sizes and molecules that were there from the ribosome down to small protein complexes. I think there were 20 classes. I'm just going to use 3 here, and then we'll have them just be orange and then green and greener. All right, but this is what you can do with VMD. This was, you put in all the ribosomes. This is more of a test result. This is not going exactly with the tomogram that I showed you. Here we just distributed as many we'd have in a fast-growing E. coli. Start putting in the small complexes. So what I wanted to show you that if you really start packing things into the cell, there you have almost like little channels being formed at any one instance in time. And now we're looking at everything that has a lattice site that is occupied. And what I mean by occupied, if you could have, say, maybe up to 8 particles, depending on their size, in a particular lattice site. If that thing is occupied, then you color it yellow, and you can see sort of the open spaces that are there. And the reaction channels are really becoming visible. All right, so I sort of like this example of how one could use BMD to get a window into the cell. Another thing that was important to do, and we're using BMD, when in the preliminary studies leading up to those E. coli studies, we looked at a typical ribosome distribution. Again, here they're in this sort of gold color. Here's a little piece of DNA. It's in red, and there's supposed to be a transcription factor, like a repressor sitting on it. And if it to come off, we were trying to see what's the effect of crowding on it rebinding to that gene. So over on the right-hand side, you see a fraction of packing, so more packing as you go over here, and you're looking at the probability of rebinding probability goes up, 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 the more crowded it is. It's just not able to escape. In principle, the 3D, as you know, 3D diffusion, it can escape and never go back to, quote, the origin where it came from. And then you can measure then in each of those cases, what is an effective or an apparent diffusion coefficient for that repressor. So I think the movie should be running. So first I show you the ribosomes, and I pack in some of the other particles. And the green and greener, right? Now I'm going to take them away so you can watch what's happening. But if it collides with one of those, then it bounces around, or it's not allowed to enter that lattice site, and it tries to escape. In this case, it does return. And as I said, it's these kinds of calculations you can use to try and come up with what are, as I said, apparent diffusion coefficients, and get at a probability of rebinding. Now, the student who did that work was Elijah Roberts. He was the student who then worked on the E. coli simulations. And we were helped by a computer scientist here at the U of I. His name is Wenmei Wu, and he is an expert on NVIDIA GPUs. In fact, he now has a joint appointment with them. But he had one of the first centers for the GPU for NVIDIA Center of Excellence was here. And he teaches how to program the GPUs in the CUDA language. All right, so now as we move over to this reaction diffusion master equation, I think it's time I really put down the equations for you. So we're talking about here the probability of the cell to be in a particular state. We're going to be looking at stochastic dynamics, at least for the genetic information processes, and maybe even some of the metabolic processes like the charging of the tRNA that involve rather small numbers. So if you talk just about the chemical master equation, whether you're doing these cell simulations or just any type of chemical simulation, you're talking about the probability of that cell to change its state X as a function of time. And this X, the state of the cell, that, it can be a long vector telling you like how many messengers, how many proteins of one type, so on and so forth. And there are going to be reactions that are going to take you out of that state, and they're going to be reactions that are going to bring you back into that state. So this sum is over the reactions that you're considering. And here, as I said, everything is well stirred. And so that couples very nicely also just to ordinary differential equations where you do not have to take the variation or the fluctuations into account. You are solving, you're not really solving, you're simulating the trajectory of this chemical master equation using the so-called Gillespie stochastic algorithm. Now, if you want to take spatial heterogeneity into account, then I need to add to this reaction term a one involving diffusion. And if you notice, I've added another sum because now each of these processes, reaction, carries a label where it is in the cell. You will impose a lattice on the system. You'll have subvolumes, but you can also say, is this a membrane reaction? Is this a reaction that's in the nucleoid region? When you do eukaryotic cells, is it one in the nucleus? Is it in endoplasmal reticulum? Is it in mitochondria? So you have an additional label to look where it is in the cell, right? And I'm showing you one of the simulations from this paper from 2011 and 2013 on E. coli. It was a genetic switch. So you can imagine you have nutriments flowing in here. If enough sugar comes in, a repressor that was sitting on the DNA falls off. And then you can start making messengers, which are red, and then they get translated at one of the ribosomes that are in the gray dots here. And it makes a membrane protein, a permease in yellow that you see diffusing all across the cell. And it lets even more sugar in. So then it switches on. And you're seeing down here the time. So this was a simulation that went on for over an hour to do this process. And this is all the experimental information we needed and the colleagues who were providing it to us. All right, so now just a little bit of pictorial picture. I'm just rewriting that evolution equation in terms of the reaction operator and diffusion operator. If we're just looking at this part, but not caring about the labels in V or the volume, the subvolume is the entire cell, that would just be the chemical master equation part of it. We add in diffusion. I'm adding this diffusion operator. So again, the blue corresponds to things also being able to hop from one sublattice to the next. And the red part indicates the reactions that can take place inside of this position in the cell, right? And this has been developed over several papers. We talk about how do you select the size of the subvolume, how do you implement it on systems with multiple GPUs, and we give further examples here in this Reports on Progress in Physics. So here again another demonstration. This is made for E. coli. Here would be my cell. The green is the outside surrounding medium. You have the membrane that's here. This would be the periplasmic region. This would be the cytoplasm. And this would be the nucleoid region. And here's where you were seeing all those ribosomes. So now we put in the particles, we discretize everything, and now we start the simulation once we defined all the reactions. And again, I'm just showing you it can diffuse from one sublattice site to the next, like here or out here, or it can react within a subvolume, right? And the time steps here are microseconds, right? And again, every time you're having to count how many particles do you have of each type in each of the subvolumes. Now, when we start connecting that with another method, like ordinary differential equations, where we really don't care, we just assume it can be happening anywhere within the cell, and we're not interested in the fluctuations and the variations in the numbers that we see. We wrote a paper in 2018 how you could select the communication times, that you look at those processes that you're treating stochastically for a certain time period, you stop, and then you exchange information to the ordinary differential equations, integrate here. And I'm showing that at that time we were using an adaptive time step differential equation solver that I think was
Essentially, it's it's not SIBO, it's L-SODA. It was developed by a colleague. Oh, I should know her name. That was had been tested for years, so that's we use. But I think any of those packages you could be doing this with. The communication time, we tried out various communication times here. Which ones had the least effect on the overall answer? And that was about 1 second. So what we are communicating are the enzyme counts, the number of nucleotides, deoxynucleotides used in transcription and DNA replication, and the recycling of the mRNA and the ATP costs, right? Okay, and one other method reaction I didn't talk that much about, although it was already indicated in the E. coli simulations. We have a procedure in which we have a sec-Y, sec-A complex that allows you to insert membrane proteins into the membrane. They are present in the minimal cell. And from work that was done by these colleagues, we know that it takes about 1 ATP per 10 amino acids to be inserting the membrane proteins at a rate at about 40 amino acids per second. So now what happens to the cell volume in those, say, let's look first at the simulations that just involve, don't take diffusion into account, or just do this, the hybrid CME, ODE. As I said, the DNA tends to double at around, make a second complete DNA chromosome at around 60, 65 minutes. And then afterwards, and this whole period here, or the growth of the volume, is dictated by the lipid metabolism that we showed you. And after that, you have an increase in surface volume until you get 2 separate cells. Now, this part is in some ways rule-based, if you want. This comes directly out of the reactions we have. And the reason I say rule-based is we do not have the cell division kinetics in there yet, involving like FTSZ, CEP-F, CEP-A, and FTSA. And so we just assume that it can form this filament here that then allows you to separate them into 2. So again, if we do these simulations over many cells, we can see that there's a variation in, and let's look here at the blue line, which would indicate when have we doubled the surface area. So we've got 2 separate cells. That can be anywhere from like about 90 minutes to 100 and maybe 10 minutes, right? And the red line gives you the average to this. Where you double the volume of this, it goes anywhere from about 60 to say 75 minutes. And you can also look at the variation in the lipid content of the surface area as you're growing over this period. The other thing that you can do is to look at the pool of metabolites to see how much it varies from cell to cell. So what we were particularly interested are those metabolites that help you make either the messenger or the DNA. And in this one cell, you see that the pool could get as large as 8 millimolar for ATP, or it could be in this other cell as low as 4. The ADP is always lower in it, and there's a lot of variation from here. And then we compare these things to this, I think, a classic paper done by Rabinovich, where he looked at the metabolomics of yeast cells, human mammalian cells, and E. coli cells. So we were doing a comparison for that and those results from E. coli. Because at the time when we first did this, this was at the beginning, we hadn't had the final version of the metabolomics for the minimal cell.
What's also interesting is the time dependence of the ATP costs. And so if you recall, when I introduced metabolism, I said, well, that thing is really important because this minimal cell has no oxidative phosphorylation. So it has to make all the ATP in central metabolism. That was this 35 to 45,000 per second. And then we started keeping track of the costs for all the various processes. Like we have active transport, bringing in like those nucleosides and the deoxynucleosides can require ATP. Bringing in other, like the amino acids, it can also require ATP. And then how about putting a membrane protein into the membrane? As we said, there's an ATP cost associated with that. And then there are ATP costs that are also in the metabolic network. You don't always make ATP. Sometimes you need to use it, right? And then there's DNA replication, and then transcription and translation. So this is sort of interesting way to look at it, but we prefer, and the top two curves are just showing you generation and total ATP costs. So we go over and let's normalize this thing to 1, the ATP costs. And we just want to see what's the fraction of each of these, because I think it gives you a better feel for it. And then you see automatically a few things just pop out. A1 was here, this DNA replication. Oh, and here, this is one that started at about, 10 minutes and was over about 60 minutes. And yeah, there's an ATP cost associated with making the DNA polymer, but it is much smaller than some of these other processes, including the active transport of the nutriments into the cell. Now, I was particularly interested in that because Stautheimer already very early on in 1973, and then later in Lynch, tried to estimate what is the cost to make a gene in a cell. And Stautheimer said very clearly that we're not going to really know the total cost until we know the costs for transport, getting nutriments into the cell. How does it do it? And as I said, we were totally, I think this is one of the first times I've seen an estimate of this ever published. So we're quite proud of that. Here's another example, cell 39 and 32. You're seeing here multiple replication events. Very interesting. So in this particular case, we didn't allow the second replication event or the forks to start forming until you had replicated the DNA once. But if you look more closely at the formula for the DNA A and the on rate and the off rates, one of them is like a second order. So it's dependent on the volume and then the particle number over the volume. So if there's a shift in how fast you're making the DNA A, as the cell is growing, you could start making a filament even on the original DNA before one complete copy is made. Right? So another interesting result that you get out of this. And the other thing we try to do is to look at the time dependence of the metabolites over a cell to see if we get approximate homeostasis. So if you have multiple replication events, that's the red line, yeah, it looks pretty good for most of the deoxynucleotides. But when you have more replication events taking place, or partial replication events taking place, the phosphate levels really start shooting up, right? And one would think this might damage the cell. And we have not put in any regulation in this model because in reducing the cell, they cut out most of the transcription factors. Anything that would regulate, say the transport of phosphate or into the cell. But it turns out there is a protein left that would regulate that when the levels of phosphate get too high. So we're fairly certain this number will drop if we impose that degree of regulation. All right, so then finally what I'm going to do is show you some results from the full 3D simulation. Remember, as I said, we're only going to do this over 20 minutes. So we are not in these simulations going to allow the cell to grow, nor the DNA to grow. So, but we can put in it and model all the metabolic pathways, and we can model transcription, the making of the mRNA, and it's also its degradation. And we can model the translation, including the insertion into the membrane. But we do not do DNA duplication in these 20 minutes. So let's look at what happens. And one thing before I go, we didn't talk much about how does the messenger decay. And in the very early on simulations, we just assumed there was a mechanism. But we now know that there are things that are called the Gratosomes. It is a multi enzyme complex, actually involves 2 of the proteins out of the central metabolism, plus some RNAs, a couple of them here, here, and here, and here, as well as a helicase. And these RNAs molecules that actually chew up the DNA, important, they can chew it up from either end, the 3' end or the 5' end, or it can start in the middle, break it into parts, and they get further degraded. So this whole complex in the simulations I'm going to show you are just going to be represented by a big red dot. So now, after the messenger is made and has been translated, or perhaps even before, there's a finite chance it could diffuse also to these degratosomes and be degraded. This is part of the recycling of the mRNA that's important in establishing the monophosphate levels that are going on in the cell. All right, so the other thing that gets varied here from what I showed you earlier, we had this plot for polymerization, like for making the RNAP acting on the single-stranded DNA to make the messenger, or this to make the protein. So now this is determined, this first step is determined by the time it takes the RNAP to diffuse to that particular gene in the DNA. All right. And that changes this formula slightly. But again, it's still there are 2 terms that are proportional to 1 over the monomer concentrations, and then the total length of the template that you're trying to copy or to make. All right, so this had to be sort of re-derived from Hofmeier's original treatment. And now the simulations, just to explain what you're seeing. You've seen this picture already of the cell membrane, and the size of it is established from cryo-electron tomography. The ribosomes are going to be in yellow, but they can turn color when they're translating a messenger. The degratosomes are going to be these red balls here attached to the membrane. And when they're active, they go from red to blue. The DNA are now orange here. And then you occasionally can watch and see a transcribing RNAP as it attaches to a gene and then reads along the DNA. And this is 20 minutes, but we've sped it up so you can watch these processes. So now you're seeing, and maybe I'll draw your attention to this area here, sort of in the middle. You're having a ribosome that's going to change color, and then shortly after, a degratosome that's sitting near the on the surface near these ribosomes will also change its color to blue as it degrades the messenger. And you occasionally see the green, you're starting to read the gene here. And then when it disappears, it's gotten to the end of the DNA. So in folding the DNA, I cannot stress this important enough. We have kept the information about where the gene is in the sequence. And if you have different conformations of the DNA in the different models, this is just one cell simulation, then these genes will then be seeing different regions of the cell. Now, what do we get out of those short simulations? Let's see, that should go on to the next one. Well, we can start looking at the counts of the messengers and comparing it over to the well-stirred models. We can do that for genetic information processing and metabolism. It looks about the same. All right, that's good. But what's interesting is you can now start out of those RDME accounts, and that's this plot here, get a distribution of mRNA half-lives, right? And I didn't show you this, but instead of just assuming a fixed half-life, say, of like 2 minutes in the CME simulations, the well-stirred ones, you can then assign a probability to each one of those. And we're trying to see if that getting the probabilistic information from the diffusion results sort of gives you a reasonably good model of the mRNA distribution of half-lives. So these half-lives go from anything from like about a minute out here to almost 15 minutes. And again, the half-lives have not been measured. The transcriptomics has not been measured for SYN3A. That's in it's being done, but it's a very slow process. But you can compare it to Bacillus subtilis and to another mycoplasma. And it agrees actually quite well with the distributions that they see there. We can also look at the scaled protein counts, but we're having to do this over 20 minutes and then compare that to what we see for the CME. You know, it looks somewhat similar. It should be really centered about 1.1 for these 20 minutes, and it's fairly close, right? And the other thing that you can start looking at is the average number of messengers, average number of translations per messengers in 20 minutes. So a messenger on average can be translated about 4 times before it decays in those 20 minutes. So you're already obtaining a lot of information about the system. And from looking at the correlations among the various subsystems over the whole cell's doubling time in the well-stirred simulations, we're now trying to abstract sort of if we can then see all the principles that people know and then even see some new emerging ones. Now, that's stuff for the science paper, and I wanted to talk to you mostly about the program today. And so this Lattice Microbes, we have used it, as I said, to not only simulate bacteria, but also eukaryotic cells. The exercise that you will do this afternoon or the tutorials is just because this is all being done Zoom. We didn't want to try and do the cell simulations with you today. Also in part because this one paper is now under review and I didn't want to upset this high profile journal. We are making the code available for 2.4, and that is much easier for a user to use. The examples, the tutorials that we'll give you this afternoon are just a sort of a sampling of what happens when you simulate some of those reactions either stochastically or deterministically, and looks at some of the early studies that were done on E. coli, which I can show you in just a few minutes. But what's important is connecting to the Lattice Microbes is all this integration of experimental information. We call that experiment to LM. It is done either in, PyLM or JLM is the newest one that goes with this 2.4 version of it. So we take omics data, we take the metabolic network, the reactions, we take the kinetic data, We take information from the geometry. If you have fluorescence data or single molecule FRET, all that is read in. And in fact, it's at this level with the notebooks is how we work with the experimental collaborators. They're very good at using Jupyter notebooks. And then one can discuss, well, is that really a dot in your fluorescence microscopy? What's the threshold? Do we count it or not? Right? And then today you'll be running the tutorial on the Amazon cloud. So we've put the program up there, and the one of my students, Troy Breyer, I'll show you his picture at the end, will be helping you to get started. Now we've set up, we have accounts for the participants that were email addresses that we were sent. But he'll have, as he starts in the afternoon session, it would be great if the majority of you could be present, because he'll get you all each up and running on your own cloud, so to say, right? And it would be great if we don't have to do that too many times as people move, come into the afternoon session. So just to get started, I will also warn you that we will close down the cloud at the end of the day, because to reserve time for over a set window costs money, and we just didn't want to have it run open for too long of a time. So it's for this afternoon session. As far as like the programmers are concerned, besides John Stone, we've been helped tremendously by Julio Maya, as well as these two students, Zane Thornburg and David Biaggi. But I would also like to point out, we are looking for another GPU programmer we would like to turn our cell simulations, when we make eukaryotic cells, into a Minecraft game. Now, I've been talking with NVIDIA, who's trying to help us with that, and in a partnership with Microsoft, but they keep telling me, I would like to just get one programmer who could both help scientifically with the algorithm plus the Minecraft. And they tell me, yeah, there's such a person, but it's called a unicorn. So it means it almost doesn't exist. Either you get a gamer or a scientific programmer. I'm still looking, so I don't know if I totally agree. So in the rest of the lecture, what I'd like to do is give you a little introduction to the sorts of problems that you can also address. These whole cell simulation, all the networks, all the reactions will be made available as soon as our paper is through the review process. And in the others, it cannot be anywhere as complete, because just a lack of knowledge of certain aspects of it. But what we try to do is take all the information that is out there, check whether it's consistent. And this also includes molecular dynamics simulations because some of that information that we get, for example, like the membrane insertion, those come out of measurements, but also through simulations. Identifying what substrate binds to a particular membrane protein also have been in part determined by experiments, but also through simulations. So there's everything that people have developed on a molecular level can be used in these simulations. All right, so let's take a quick look. This is on a simulation of an E. coli cell. So as I said, what we want to look at is ribosome biogenesis. So we have a messenger, say, coming from a gene. And of course, it can diffuse. And then eventually that messenger will associate with a small subunit. And now and a small subunit, makes and a large subunit makes a ribosome. And you read through the messenger up there. And then things fall apart. And the 16S is produced at the site of where the rRNA operon is, this started up a little bit fast. So let me go back and explain first. Now we're having these orange balls just indicate genes or operons on the DNA. So let me try to go back here and start the thing up again. And let's see if it does this. So there's our simulation box for that E. coli. So I want to look inside into the nucleoid region. All right, so the operons are localized to sites within the nucleus, and then you have maybe a messenger being formed. It, of course, can diffuse. This messenger can bind to the small subunit, and then it associates with the large subunit. And that is a picture of an intact translating ribosome. And then up at the top, you're seeing it read through that operon, which contains the genes, say, for several proteins. It falls apart. And what you're supposed to be thinking is, oh, that was reading one of the operons where you have many of the ribosomal proteins sitting there. And now you start the assembly process. One protein after the next, and those are now these little gray balls, have to start assembling. And that blue thing turns bluer and bluer until you have an intact small subunit that has about, the 16S and 20 ribosomal proteins to it. And then you close it up. Now, if you noticed in this picture, it treated the assembly of the ribosomes. And we were lucky when we were doing that study, we had the help of one of the leading experimentalists for ribosome assembly, Jamie Williamson, out at Scripps. So we could parameterize the kinetics of the binding of each of the ribosomal proteins to the rRNA and build this model. But what we didn't allow is for the cell to grow, and we didn't have a full metabolic model in this at this time. So we at least tried to address the next problem. Well, could we look at the same set of simulations, but now allow the cell to grow? And then what predictions could you make? So again, we're not putting in metabolism at the moment. We're just looking at ribosome biogenesis. So you're forming the messengers for the ribosomal proteins and for the rRNA. And then you're just looking at the assembly, but you're allowing the system to grow. And the reason we knew how to make this thing grow is another set of experiments that were done from a colleague who labeled like 14 different places on the E. coli. And he just saw when you saw each of those 14 places being duplicated, and he measured it in time as well as the size of the cell. So he took that information and imposed it onto this cell simulation. So it wasn't grown by lipid metabolism like we do in the minimal cell. It was grown by the rules that we saw from experiments. And now you're going to see some of the intermediates pop up in different colors, because as you're forming the small subunit, you have what are called the primary ribosomal proteins bind directly to the RNA and start decreasing the radius of gyration. Then you have the secondary and the tertiary. And we're interested, where do you find these things at the end of the day? All right, so let's let it rip. So first thing you see is you're starting to see the DNA duplicated. So it's over here on the right side as well. And then everything's are popping in and out because they're disappearing and being made. And then voila, now you have a second cell. And you do this often enough and collect the statistics, you can make predictions about where you see the intermediates. And that was, oh, let me go back. I didn't put that plot in here. But in this paper, we present that information. And it turns out, this was in 2016. I think in 2018, someone at Johns Hopkins started to measure labeling those intermediates. and they could start seeing them generally in the region that we had identified. Now, let's go on to the next application. This is on trying to do stochastic dynamics and integrate those with the tomograms, but we do it for yeasts now. So the tomograms are coming from Elizabeth Vila and Wolfgang Baumeister, and we will then later on look at a HeLa cell, and there the tomograms were coming from Julia Mohammed. At one time, Julia and Elizabeth were both group leaders in Wolfgang Baumeister's lab. They now have independent positions. Elizabeth's at UCSD and Julia's in Heidelberg at the EMBL. So again, you start with the tomogram and panel A. It's sort of hard to see things, so they do their reconstructions. But what you see over in panel B is this big nucleus in the middle, and the little purple in the upper right-hand corner is actually the edge of the cell. So the nucleus is sitting not in the center of the cell, it's sort of, shoved off to one side. And then the rest of the fragments you're seeing are the ER and the endoplasmal reticulum. Now, if you take that slice that they made of the yeast cell with fib milling and rotate it on its side, you can now see the holes in the nuclear membrane, the nuclear pore complexes, where the mRNA will come out and then be translated by ribosomes. They're colored blue here. Now, some of them will be embedded in the ER, but some of them also will be free. And from this information, then Tyler Ernest, who you see down here, who was the lead author, started to reconstruct a whole model for the yeast based on the tomography data. And let's build it up. Here's from a slice, add more, and then now you have sort of a picture of what that cell could be looking like. So this beige part would be the nucleus. There would be a huge vacuole that's here, and these would be some of the mitochondria. The ER was too convoluted in there for us to build an accurate model. And as you know, the ER would help transport particularly membrane proteins that are made on the ribosomes embedded in it to its positions, various positions in the cell. So we just have it diffused. So we have to define sort of a diffusion coefficients from those for those objects. So what we wanted to do is look at a particular experiment, see if we could, with this model, reproduce some of the results. And what we wanted to look at was a hybrid RDME, CME, ODE. So as I said, this was the previous attempt when we had a bit of the metabolism in there. So we were doing the ODE, and we had genetic information processing in there, in that what we're looking at is having galactose come through, being imported through a transporter G2. It can be partially metabolized, or it can bind with transcription factors, or actually repressors. And when it binds to it, it can no longer get into the cell. So these genes that are being repressed suddenly turn on like a switch. And one of those genes is for G2, this transporter. So you can make more transporters, and then more galactose can be brought in. As you see on the outside, there's 11 millimolars. You'll never reach that exact concentration on the inside, because as I say, some of it is being metabolized, but it can get up to as high as maybe 5 or 6 millimolar inside of it. Now, this had been well studied in that the kinetic equations were provided by Ramsey and his group and Nature Genetics. Some of the single molecule studies had been done by Uden Arden in Nature in 2006. So we had a lot of information we could use for comparison and to set up the kinetic model.
And in this paper, what we're primarily interested in is looking at what should be the communication times between the ODE and the CME part, which is inherent in the subvolumes of the RDME. Okay, so let's take a look. And again, when we solve the chemical master equation in a subvolume, we assume it's uniform within the subvolume. And we have differential equations handling like the transport and parts of the metabolic network. And here were the communication times we talked about. So here's a quick movie of what's going on. So this is the cell that was made from Tyler Ernest's work in 2017. And now inside the nucleus, you see sort of these little holes, and there's supposed to be a gene, and it's off at the moment. Then the repressor will fall off, and that gene is going to code for more of the G2 transporters. And when it diffuses out of the nucleus through these pores and gets translated on one of the many ribosomes, you see it as yellow. And then it will diffuse to the membrane. So you see them accumulating here along this membrane. It's in blue. And over here, we are watching this process over about 60 minutes. And there are two plots here. And I'll go through once again. So what you predict out of this is an asymmetric placement of these transporters. And now if we can watch the movie again, but look in the corner plot, you're doing two things. You're looking at the concentration of the galactose. As I said, that's going to be in green, and we could assume in the cell initially there's none, and it gets up to about 5 millimolar. And with the transporters, we know that there's about 1,000, and at the end of the run, it already had gone up. Please see this as times 10 to the 3. It's about 15,000 of them. So it went from 1,000 to 15,000 of them in the time period of about 55 or 60 minutes. So we looked at the results and compared them. They compared rather well, although I will tell you a funny story with respect to this work, that when we were testing the communication times and the accuracy of the methods, it was obviously a computer scientist who came in and he wanted another plot, and the journal was limiting us to a certain number of plots. So he asked us to take away the comparison to the experiments to show another accuracy plot. And we go, no way, we'll put that newly requested plot into the SI. And what I'm showing you here are the so-called performance pictures that we have for this system. It's not a lot of reactions, just like 75 reactions compared to the 1,000 or more that I was showing you for the minimal cell. But if you want to simulate the cell, say, for a day, then you better use the better GPUs, like either 4 or 8 of them. There you get almost an hour and a half of the cell cycle in. If you're using like a Tesla K80 or any of these other ones here, then you have, of course, less good performance. So it does really depend on your most recent GPUs, and now their VT100s are out in the NVIDIA box, and again, we get a boost in performance. Another issue or point I want to make clear, this is now from the work of Julia Mohammed and Elizabeth Vila when they were group members with Baumeister and appeared in Science. And they were trying to look at the molecular sociology near the nucleus of a HeLa cell. That's a human cancer cell. And right now, this black area where I've written all the legends, that's the nucleus. And these are these nuclear pores, and they have them in beautiful resolution. And in fact, the top part of this plot is Julia's data. And this shows you in the bottom part of the plot, below the dashed line, what happens to this when we put it into our cell simulations. And so now it's determined by
the size, the lattice spacings. What is the size of our subvolumes or our sublattices? And again, here makes it even clearer. Here's what they see, and this is what we would see in our simulations. And then what we did was we tried to simulate things coming in and out of these pores, because again, that was just beginning to be measured, and we could do those studies, and that is a part of that study in that I showed you, where Tyler was doing not only for yeast but also for the HeLa cells. All right, and then finally, more recently, we took the sort of models that we had information from Julia, and now there are more information available about how many mitochondria are in the cell, how much of the cell volume is taken up by the ER, and we could try to build an even more realistic model of the HeLa cell. And what we were interested in this paper that appeared in PLOS Comp Bio last year was how try to just follow the process of mRNA splicing. So you get because of the intron exon structure of the genome, you have to remove the introns when you're making the final mature messenger. And it is known that part of that splicing procedure involves having things go in and out of the nucleus as part of that cell cycle. So in this study, we were trying to study that. And also one is beginning to identify those organelles, those membraneless organelles that are in the nucleus that are involved with splicing. So they are called speckles. Just exactly what's in them, what part of the splicing mechanism and the spliceosome is in there, still being determined. It's just like the nucleolus. People know that that is sort of like a collection site for making of the ribosomes. And these are all part of these condensates that people talk about, and people are actually trying to actively determine what's in them. And in the case of the splicing, correlate it with positions on the chromosome where a gene is being actively transcribed. So perhaps let me end the lecture with just reviewing why one needs this lattice microbes. I think structural biology in both the cryo-EM and molecular dynamics, they're really revealing the structural biology of parts of this, of all the different components that are involved in the whole cell. And we're also having a real revolution in the omics, getting at what's in the cell. And some of it is even being done at a single cell level. This is from the work of the pioneering work of Sunny Shi, when he was looking at the protein copy number in maybe like 300 E. coli cells. It's also been done for yeast, and it's beginning to be done for stem cells. And as I said, from the pioneering work of Ilowitz, we know that the genes, that gene expression is stochastic. So that means the numbers are going to vary of the messengers and even of the proteins at any one instance. So sometimes people say, oh, it's like you'll have snowflakes here. They'll cause phenotypic behavior. And then if you now start getting a good model of the cell and all of its components, you can start addressing how do the cells react to drugs or mutations in the reaction pathways. So one of the things that we're hoping with the minimal cell is that you'll be able to add reactions to it, complexify it. It has dramatically reduced in the metabolic pathways, but we know other pathways are there in other organisms. introduce them and see how the system responds to it. So we really do feel that the community starts needing stochastic dynamics, but at a cell biology at the scale of an entire cell. And I think it's only fitting now just to remind you what you have seen in this workshop so far is biological modeling at multiple scales. So we can go from atoms all the way up to single cells and start looking at multi-cell organisms or populations of cells. And each one of those methods that you've learned about, like with VMNAMD, are fantastic at these different scales. So if you want to look at atomic interactions, there have been years and decades of work done on those intermolecular potentials, force fields for MD simulations. I think there was also a Nobel Prize given for that work, and is developing these force fields for biological systems. So you can look at, a protein being made at the ribosome, maybe that's a transcription factor that can then come and interact with the DNA. Those are all types of simulations that you can do. And you can look at rather large assemblies. The main problem that one has when you stay at the all atom level are the ones at the time steps that you're sort of restricted to femtoseconds. You can try to improve the situation by doing rigid body interactions. Just look at like large subunit, small subunit, but then you have to have a potential base, coarse grained. You have to modify these potentials, right, to represent this rigid body. And then when you're over to the RDME, we're now back at the level of it's probability based. So you have to know something about the reaction probabilities, probability for diffusion. So the diffusion of proteins in cells has been measured. You have sort of a rough size dependence. The same way on the messenger, you can use the hydrodynamic radius for that. There have been enough measurements on mRNA diffusion that you can start making estimates of what their diffusion coefficient should be. And that also goes into the cell simulations. These time steps are moving more into the picoseconds. And as I said, ours are into the microseconds. And it is completely GPU based and also uses VMD to look at it. Although the movie on E. coli, that was done with Blender, so we could piece together bits and parts of the simulation. And then also bring in the whole ribosome, because I think it's really going to be important as one of the final validations is to go back and do a complete physical model of the minimal cell, which was something that my husband had wanted to do before he passed away. And I'll tell you a little bit about how that effort is proceeding, but now instead of using the all atom force field, using a more coarse grained force field, the Martini force field by Mimaric. So as I said, each of these length scales has its own time scale associated with it and a length scale. And what I've been talking about is mostly this region over here. So my things are like half a micron or micron or more in length here. One thing this plot cannot do is also bring in the notion of the dependence on the concentrations of particles in there. So the stochastic treatment is more important for reactions involving particles in rather small amounts. If you get into certain metabolic reactions where you have millimolars or half a millimolar or 10 millimolars or something, the fluctuations in those reactants is not critical, right? So you have to be able to combine these things with ordinary differential equations. And then one can also start combining it yet with yet another methodology called like Brownian dynamics. If you want in the cell simulations, the DNA also to move as you're doing the simulations. All right, so that was, first attempt was published in 2018, and a lot of these different methodologies are introduced through what we call hook simulations. So again, they communication time to passing information, and sometimes some of the control of that is also done with this JLM notebooks. One thing I didn't talk about, but you'll be hearing about it from Raphael Bernardi, is that in 2018, we added another component to the NAMD. That was the ability of it to do quantum mechanics. So I think the title of the paper here was NAMD Goes Quantum. And we wanted to look and have the ability to look at certain of the metabolic reactions. Like what I was primarily interested in was the focus of this paper was the reaction that sets the genetic code in a cell. It's the charging of the tRNA with the correct amino acid. And I think I have a little movie from that paper that can show you both the power of the technique, but also the real power of VMD being able to show you molecular orbitals as a function of time. Okay, in fact, that's the next movie here. So I hope if I just click on this thing, it will start running. So we're going to look at the charging of the tRNA, and you're going to see this is done with the help of these proteins called aminoacyl tRNA synthetase. This is the one that loads glutamine or glutamate. I didn't look quickly enough. And here's the anticodon. And here's the region of the protein interacts with it. But information has to be communicated all the way up here to the active site, because the amino acid is put on the CCA end of the tRNA. So let's zoom into that active site. And we're looking at sort of this last step of where you're dealing with an adenylate. So the glutamate has already reacted with an ATP. So you have an AMP moiety connected to it and the amino acid moiety. And here's the final CCA on the tRNA. And what has to happen is the amino acid has to go over here onto either the 2' OH or the 3' OH. And what I'm trying to show you here are the time changes in the highest occupied orbitals. And in a few seconds, this proton from the OH will go over here, and then the amino acid will be attached there. If you blink, you'll miss it, but whoop, there it went. So now it's over here, and the amino acid is connected here. And what we did in that Nature Methods study, we did this entire reaction, but then we also looked at how long it took this AMP moiety to get out of the active site. And that takes a long time. And but you can calculate the barriers that you have for that process. Now, I'm pretty sure Rafi will show you a different metabolic reaction, because he's gone on and to apply it to many more systems, things that interest him more. He was my postdoc at the time, and Marcelo Mello was a graduate student. So and this was part of Marcelo's project, was to look at the charging of the tRNA. So that's why we focused in on that aspect of it. But what's also equally important is the choice of the quantum mechanical simulations you use. So they wrote an interface where you could bring in many different methods, whether it be ORCA or some other some other method that people use. It's all allowed through the interface. And now what remains to be done is to comment a bit on the people who are involved in this. So you really do need a big group of people, to help do the experiments to validate what you're doing, and also to help work on the algorithms. So I think I've mentioned most of these people as we went on. These are the students that are involved. And I didn't get a chance to talk about the work that's going on with Marek because it's still in just its beginning. But we, as I said, we do hope to have at least a coarse grain model at the level of a Martini 3 version of the minimal cell, say within a year. That's our goal, is to do that. But otherwise, these are, Rashid works with Ramos Dane at Leiden, and they're the ones who do the chromosome capture maps. Here's Elizabeth Vila and Wolfgang Baumeister. He's still the Max Planck director in Munich. And as I said, Elizabeth is in UCSD. These group of people over here are part of the synthetic biology group. This was my postdoc, Miriam Breuer, who led the work to establish the essential metabolism. That work enabled him to get a very nice position in the Netherlands. He came from Germany, so he's close to home now. And Tyler Ernst, along with a couple other of my students and my main programmer for many years, started a startup company called Sim Biosystems and are doing quite well. And then this is the colleague from computer science. And of course, we deeply appreciate the funding from not only the NIH, but we also have on campus an NSF Physics Frontier Center for the Center for the Physics of Living Cells. And the international network of students needed for this project is also funded by the Physics of Living Systems, the POLS project. So we are also extremely lucky to have a really collegial interactions with NVIDIA, of course, with NCSA, and a grant from the Keck Foundation. So with that, I'm a little early, but hopefully it showed you what one can do with this. And now I think we'll go ahead and stop, and I can start entertaining any questions that you have or comments or information that you'd like to have about these simulations. Okay, if there are none, what I would say, Troy, are you connected? That'll be the teaching assistant. So let me show you. Well, let me put this back up. There's a picture of Troy.
Yes, I am connected.
Yeah. So can you? I don't know if you're allowed to do screen sharing. Do you have the tutorial, the PDF of the tutorial that you could bring up? I forgot to put it in here, just to show them what they're going to be able to do this afternoon.
Yeah, 1 second.
So it could be that Mariano has to give you permission and I can stop sharing.
I think he should be able to start sharing.
Yeah, I can now. 1 second. So what you're seeing right now then would be an example of the tutorials, and I'm going to go through this a little bit more more systematically during the actual tutorial hour, but it'll be run through Jupyter notebooks. And so this is an example of one of the tutorials where there's some description about kind of what you're going through and what you're doing. And I'll also give some background on some of the information in the tutorials. And so hopefully from this, what you'll be able to do is get an idea of how to run a simulation using LM and then some of the capabilities of LM. And as Zan said, this will all be done through the AWS, so it'll be available for today, but after today you will not be able to access this anymore, but I'll have some additional information on what the outlook would look like being able to do it at a later date or on your own machine if that was something you were interested in.
Now, if you are interested, in running these, I know you guys often come to these workshops having other projects that you're trying to work on, and that's fine. But if you, I think the afternoon session starts when, Troy?
2 o'clock.
2 o'clock. So at 2 o'clock, Troy will be there, and he has your email addresses. So how do you want them to communicate that they would like to have an AWS account?
So I can see the number of people that are in here now. So I'm just going to launch a certain amount, and then during that tutorial hour, the tutorial period, I'll just show my screen, which will have the IPs for people to connect, and I'll explain that a little bit more during the tutorial session. Essentially, each person will be assigned their own kind of instance, as they're called, to be able to use. You can kind of do whatever you want on it. You don't necessarily even have to worry about messing anything up, due to the fact that once they're terminated, they're essentially reset. So it'll very much give you a chance to be able to kind of see some simple examples, but then additionally, you can modify and play with the tutorial programs to kind of add in some additional kind of concepts or ideas. For example, Zan touched upon the gene duplication having an impact. That's something that can easily be implemented into the simulations, and I'll kind of talk about and show ways of doing that.
Okay. Yeah, so I'll be there briefly at 2 o'clock. If there are any questions you think of over lunch and you'd like to have addressed. As I said, I know this is really the most exotic of the methods and that we presented today. Whereas normally when you're doing molecular dynamics, you worry primarily about having an X-ray structure that is, and it's somewhat complete, and then trying to make a membrane, perhaps, and insert it into a membrane. But here we're, there's a lot of information, but there is also, there's required, but there's also a lot of information out there. But as you move forward, you may work on the protein from a particular organism. And as you know, they don't have X-ray structures of proteins, even of great interest, that are from all the organisms that you want to work on. And so one of the things that also this protein structure prediction phase that went on for many years is just the ability to generalize your results, right? Can you make, if you have a structure and the sequence is somewhat similar to the sequence of that protein with the structure, can you build a model and use it? And the answer is yes. And in fact, I think Raphael Bernardi is working to have a interface of VMD to AlphaFold. So AlphaFold was a Google effort that I think was like the very best method to do protein structure prediction in the most recent critical assessment of protein structure prediction algorithms. So that is there to also help you to do it. But when you get to the host cell, it's, as I said, you're really generalizing the information. And one of the things we want to do is take a state of the cell that we have, use information about structures, like ribosome structures are known. That was a Nobel Prize. Is put them in where we have our ribosomes and then back transform it to an all atom or a coarse grain model to see if those states are reasonable, right? Okay, if there are no further questions, then I would suggest we break for lunch and then regroup at around 2 o'clock.